webcast is now starting. All attendees are in listen-only mode.
All right. Good morning, everyone. Good afternoon, some. Hello. Thank you so much for joining our live webinar today, which we have titled, How Long is Too Long? So determining the need for time and temperature controls in food handling and batching. So I'm Scott Mishakian. I'm with Eurofins Marketing, moderating today's session. So joining me as our featured presenters are Martha Kimber and Shirin Ab. Martha and Shirin are senior technical managers as part of our advanced food microbiology business at Eurofins, operating out of Fresno, California, who perform food safety research projects both domestically and internationally. All right, so before I begin, I want to relay a few general notes on the webinar structure. So of course, we're recording this webinar. Many of you will ask that the slides and the presentation, the recording, will be emailed to you within 3 days after the completion of the webinar. So be sure to look for that. One last thing is during the webinar, you can submit questions. We'll likely not answer questions throughout the webinar, and we'll hold off for Q&A at the end. In order to submit questions, you all should see in the GoToWebinar sidebar menu that will display a few options on there. One of those is the questions. What you do is just kind of type in your question and hit the enter key, and that'll go into a queue. If you have some technical issues, you can submit those through that sidebar menu as well, and we'll be sure to address those as we go through. All right, so with that short introduction, Martha, I'll hand this over to you to begin the conversation.
Thank you, Scott. The title is How Long is Too Long? And Shirin, next slide, please. So quick outline here. We'll talk about the big picture. Why is this all important? We'll talk about temperature control for safety, product assessment considerations, challenge study considerations, and then we'll switch gears a little bit and talk about the batching procedure and whether it might increase your micro risk. And finally, we'll finish off with a few case studies. All right, so big picture. Does your finished product support the growth of pathogenic organisms? I have a few different items pictured here, just as examples. So the first would be like a garlic bread and a pumpkin pie that you might find at the grocery bakery aisle. And then I also have a non-insulated beverage dispenser just to represent beverage products that might be held at ambient temperature in a non-insulated container, and whether things might be growing during that holding step. We're also going to be talking about the batching process. So does your batching process allow for growth of pathogenic organisms? There's a little nuance there. This doesn't exactly fit the definition of TCS, but they're very similar concerns. And then represented here, I have some plant-based nuggets, and then also a stainless steel batching tank to represent batching of liquid beverage products. All right, get started. We'll talk about temperature control for safety, or TCS. So TCS is necessary when the process or handling of a product provides opportunity for contamination, and the intrinsic parameters of the product, such as pH or water activity, are permissive of pathogen growth and or toxin production. And most of what we're going to be talking about here is based on a report from the IFT task force and its evaluation definition of potentially hazardous foods. In some other guidance documents, they call this time temperature controls for safety, but in this document, they recommend calling it temperature control for safety. So in interest of brevity, we're just going to say temperature control for safety moving forward. All right, and when you're looking at these products, there's a few things that you want to consider. So the intrinsic factors that can influence microbial growth can include pH and water activity, the ingredient composition, and also the nutrients that are present in the product. Also antimicrobials, you have sorbate or benzoate, for example, and also maybe there's competitive microflora that might outcompete growth of the pathogens. You also want to consider the extrinsic factors that influence microbial growth. This might include processing steps. For example, maybe your process will kill vegetative pathogens for a pasteurization step, but it's not high enough to kill spores. So that would be something you would want to consider. You'd also want to look at the packaging and the packaging atmosphere. So if you have modified atmosphere packaging, for example, that may bring some anaerobic organism into play that you would have to consider more closely. For example, Clostridium botulinum is a strict anaerobe, so it's of concern more often in modified atmosphere packaging. You also want to consider the time and temperature use period. An example here might be at a restaurant where they keep the cheese out at ambient over an entire shift. And that's just because it's easier to get that nice melt on the sandwiches they're making, and also just for production efficiency. And then finally, we want to consider the storage temperature of the product. So as an example here, we'll talk about low acid beverages and whether they might be considered TCS foods. So again, I have the little picture of that non-insulated beverage dispenser. If it's low acid, you know that the pH is going to be greater than 4.6. So that would be the right column. And then because they're beverages, most likely the water activity is going to be quite high, close to 1. So that would put you in this bottom right corner, which is a question mark or maybe. It might be a TCS food. This flowchart is from that potentially hazardous foods document. And it's just kind of looking through the different steps you might take to decide whether a product is TCS or non-TCS. So first, we need to make that decision, and we determine that it's a maybe for that low acid beverage example. The next step would be to perform a product assessment. This table is from the National Advisory Committee on Microbiological Criteria for Foods, or NACMAF group. Their documents entitled Parameters for Determining Inoculated Pack Challenge Study Protocols. I find this table really useful in determining the organisms that might be capable of growing at different pH and water activity combinations. So again, if we're looking at this low acid beverage, the pH is going to be greater than 4.6, so on this right column, and then the water activity is going to be high. And then it's going to circle here the different groups of organisms that might be pertinent. And I know this is small, so I'm zooming out in a larger format in the next slide. So these were the organisms that were pictured in that right column. So Bacillus cereus, proteolytic and non-proteolytic Clostridium botulinum, Clostridium perfringens, E. coli, Listeria, Salmonella, Staph aureus, and Vibrio. The organisms that are in bold are toxin producers. And a quick note here, Campylobacter, Shigella, and Yersinia are not listed here because they're typically controlled when the other pathogens listed are addressed. And that's a note from the NACMAF document. And I do want to call out that Bacillus cereus and Staph aureus can produce a heat-stable toxin that is not destroyed by thermal processing. So this is really important for the batching step as well, because you don't want toxin to be produced during batching that can survive your thermal process and then cause illness later on in the final product. All right, so once you've done your product assessment and you've thought more about the intrinsic and extrinsic parameters that can allow for growth, you've identified your challenge organisms, the next would be to do a challenge study. So that's the next step. And here are a couple different challenge study considerations that you would think about. So the inoculation step. First off, we want to select pathogens based on your assessment. Usually you want to pick at least 3 to 5 strains that are associated with similar products or outbreaks. Your inoculation step should not alter the pH or water activity of the product. And finally, you want to target all components of the product itself. So if we're thinking about, say, a donut that's filled, we want to make sure we're getting the filling part of the donut the interface between the filling and the outside crust, and then the crust. You want to make sure we get each different component inoculated. In terms of product formulation, you want to select your worst case scenario formulation in terms of pH, salt, preservatives, etc. Or if it's difficult to choose a worst case, you might want to test multiple lots of product. And this is because you want to observe if there's any variability. And finally, your challenge study duration and storage conditions, you want at least 1.3 times your desired shelf life. If you have a very short shelf life on the order of a couple of days, for example, like with bakery products, you might even want to target 1.5 or 2 times your desired shelf life. You also want to make sure that the samples are in representative packaging for your product package, and that the storage temperature makes sense for what you're doing storage in the finished product. In terms of data interpretation, you need to make sure that you have enough sampling times and data to show a trend. So for example, we want to know, are populations increasing? Are the populations stable? Are they dying off? And then finally, the study needs to be designed around whether they can answer this pass-fail criteria. So the criterion for the vegetative pathogens E. coli, Listeria, and Salmonella are less than 1 log of growth over the duration of the study. For the organism group Bacillus cereus, Clostridium perfringens, and Staph aureus, it's less than 3 logs of growth. And the reason why that's a higher level is because they need to grow to a quite high level in order to cause illness. And Bacillus cereus and Staph aureus have an asterisk here because they produce toxin when they grow to high levels. And then for Clostridium botulinum, non-proteolytic and proteolytic type, the criterion is an absence of toxin formation. All right, so after you've done your challenge testing, the ultimate goal would be that you choose or you determine whether the product is TCS or non-TCS based on the results of the study. So if the criterion is that there's no toxin produced and you see toxin is produced during the target shelf life, then the product should be temperature controlled for safety. Now I'm going to switch gears a little bit and talk about batching procedure and whether it might increase your micro risk. So we know that batching is not covered by TCS because this is in process and not relevant to the finished product. However, many of the same considerations are used to assess food safety risk. So again, you want to think about the intrinsic properties like the ingredients, the pH, the water activity, and also the extrinsic properties of the product. So whether it's being held at a certain temperature or if there's exposure to the environment. And this is where I want to call out again that it's important to note heat-stable toxins are produced or can be produced by Bacillus cereus and Staphylococcus aureus, and they will not be addressed by later processing steps. So we want to prevent growth and toxin production by these organisms during batching. Some examples of these products where these considerations may be relevant might be low acid beverages and plant-based foods. A lot of the flours, gums, protein powders, etc. have a larger particle size and they might be difficult to mix or incorporate. Some of these ingredients require hydration of these dry ingredients at elevated temperature for an extended period of time. Also, some of the ingredients have very high spore loads, so that's something to keep in mind. And then finally, for these plant-based nuggets and sausages, things like that, there's often a molding or forming step, and that can occur at ambient temperature, which may allow for growth. So just knowledge is power. Knowing where your risks are is helpful so that you can control them. And these novel products and processing methods may introduce new micro risks. All right, now I'm going to turn over to Scott.
All right, thank you, Martha. I'm going to put up a quick polling question as we before we transition over to a little overview on the case studies here. Let me go ahead and load that up for you all just to get a idea here. You should see. So are you concerned with issues in batching, issues in finished product handling, or both? So it's a little interesting here to get a feel for where we're at with the audience. And as we get into the case studies, we'll talk about a few of these examples. Should help out some. So I'll let that go for a second. Martha, I hope you don't mind me asking a quick question. I was curious about challenge studies when you talked about not ensuring that you don't increase the water activity.
Mm-hmm.
How do you do that? I'm picturing like you make like a cocktail of whatever organisms and it's something, but like, do you have like, does your model correct for the increase of any liquid, or just really concentrate it, or maybe something else?
Yeah, so first off, we try to use a very small amount of inoculum, so usually less than 0.1% of the total sample size when we're inoculating. We also have methodology to decrease the water activity of the inoculum itself using glycerol. So let's say that your product has a water activity of like 0.8. We could decrease the water activity of our inoculum as well to 0.8. And then that way, when we're adding it, it's not changing it.
Interesting. Okay, so you just kind of like put a correction in there to match it.
Right.
Mm-hmm. Nice. Okay. I was wondering about that. Thanks.
Sure.
All right, looks like we got some good responses and nothing's really changing. So I'll go ahead and close this, kind of give you a feel for where we're at, share where we were at on that as well. Pretty good mix. A lot of people concerned about both here. And then if you kind of split it up a little bit and perhaps a little bit more on the batching there. So, all right, well, I'll transition this over to Sharon, hopefully complement some of Martha's presentation with the series of case studies and give a few examples with what we're talking about here. So go ahead and hide this, I believe, and Sharon, I'll let you go ahead and get started.
Okay, thank you. Thank you, Martha and Scott. Here in this part of the presentation, we will cover examples of the challenge study to determine the growth of selected microorganism in the finished product or during the batching step. The example that we will cover here, including an example for a low acid beverage, 2 examples for bakery items, and 1 example for plant-based beverage during a batching step. I would like here to emphasize that, let's see how I can go to emphasize that data presented here is for demonstration purpose only. Please do not apply the information to your product and process. Example number 1 here is about a low acid beverage. Examples can be cold brew coffee, tea, or nut milk. For any batch of product that we receive for an experiment, such as a challenge study, we will test the samples for analytical and microbiological analysis. In this example, we tested the product for pH, water activity, TA, and Brix. And also for microbiological analysis, samples were tested for total plate count, anaerobic organisms, spoilage organisms such as yeast, mold, and lactic acid bacteria. Here is a quick overview of a study design for this product. Target organisms were divided into 2 groups, spore former organisms such as Bacillus cereus, proteolytic and non-proteolytic Clostridium botulinum, and vegetative cells such as Listeria monocytogenes, pathogenic E. coli, Salmonella, and Staphylococcus aureus. The objective of the challenge studies was to determine if growth and toxin production of target organism happened over the storage period. Therefore, relatively low inoculum level, about 2 to 3 log CFU per ml, were targeted for the challenge study. Basically, we are looking at a growth study here. The sample were stored at 25 degrees C. Duration of the study was 1.5 times of the shelf life of the product. For example, if the shelf life of the product expected to be 2 weeks, The experiment duration of the experiment was 3 weeks. 6 to 7 sampling times were tested over 3 weeks. Number of replicate was 3 or 5 replicate sample per sampling time. And the experiment was done in 2 independent trials. Basically, we repeated the experiment twice. The results of the challenge study are presented here. The result of each graph is presented in a separate, for each organism separated in a, presented in a separate graph. Y-axis is the population level and X-axis is the incubation time. Trial 1 and trial 2 results are presented in solid and dashed line. As you see here, the initial level of inoculum for all organisms were about 3 log CFU per mil. And when we look at the trend for the survival of the vegetative pathogen, we will see that E. coli, Listeria, Salmonella, and Staph, the population level declined to below the limit of detection after a few days of incubation. Possibly some antimicrobial compound presented in the product which killed the vegetative pathogen organisms. Let's review the result for Bacillus cereus together here. Again, initial level of inoculum was about 3 logs, but we see that population of Bacillus cereus was relatively stable over the storage period for 21 days. And the difference is Bacillus cereus is a spore former organism, as spores tend to be more resistant into environment and harsh environment. And the population did not die, but still we did not observe any growth for these organisms, spore formers, during the incubation period. Here, the results for the toxin testing for Clostridium botulinum are presented. Protolytic Clostridium botulinum sample were tested for toxin A and B, and non-protolytic Clostridium botulinum sample were tested for toxin types B and E. As you see here, toxin was not detected in any of the inoculated sample over 21 days of storage under anaerobic condition at 25 degrees C. Here is a summary of the results. Pathogenic E. coli, Listeria, and Salmonella, less than 1 log growth, or actually they died off. The performance criteria met. Bacillus cereus and Staphylococcus aureus, Martha earlier mentioned, the performance criteria is less than 3 log growth. That was met. And for protolytic and non-protolytic Clostridium botulinum, toxin was not detected. All performance criteria here was met, and therefore we can say this product can be considered as a non-TCS food. Let's look at example number 2 again. Here is the bakery items. I have 2 examples here. Bakery items can be products such as pumpkin pie, cheese, Danish, cream-filled donuts. And again, for each product that we receive, we tested for analytical here for pH and water activity and microbiological analysis for total plate count and spoilage organism. I would like to take a minute here and present how important it is when we are measuring the pH and water activity of a sample. What is the correct way to measure that? Here you see a slice of pumpkin pie. And we see different components here is like filling, crust, and interface between the filling and crust. When we measure the pH and water activity, we measure each component separately. We don't mix the sample to measure one water activity and say this is the water activity of sample. It's very important to measure each component's pH and water activity separately. Overall study design is presented here. Target organisms for this experiment was Listeria monocytogenes, Salmonella, and Staphylococcus aureus. Again, we are looking at the growth body, and inoculum level was 2 to 3 log CFU per gram. Inoculation site, Martha mentioned that as well. When we inoculate the sample, we make sure that we deliver the inoculum to all components of the product. To the filling, crust, and interface, There are different methods to do that. One way of doing that is using needle. We use sterile needle to deliver the inoculum to different components of a sample. Storage temperature, again, 25 degrees C. Duration of the study, 1.5 times of the shelf life of the product, 6 to 7 sampling times, and number of replicate was 3 samples, 2 or 3 independent trials were completed. The first item that I'm presenting the results here had a shelf life of expected shelf life of 4 days. Therefore, the study was done over 6 days. The graph that you see here, it is a population for Salmonella for all 3 trials, 1, 2, and 3. As you see, all results for the 3 trials were very similar, and Salmonella population pretty stable over the 6 days of this experiment. Performance criteria met for Salmonella since it's less than 1 log growth. Listeria monocytogenes is very similar to Salmonella results. Its population were stable, less than 1 log growth were observed, basically no growth. Looking at Staphylococcus aureus, here we see that population grew between day 5 and 6. If we look at the initial level of inoculum was about 2.5 logs, and then the population at the end of day 6 was about 3.5 logs. Even though there was a growth, but still it met the performance criteria because for Staphylococcus aureus, the performance criteria was less than 3 log of growth from the initial inoculum level. Therefore, over 6 days of storage at 25, the bakery item tested in this project did not support the growth or toxin production of the pertinent pathogen identified for this category of product. And this product can be considered as a non-TCS food. This is the second example for the bakery item. The expected shelf life for this product was only 24 hours. Therefore, the experiment was done over 36 hours. The result for Salmonella here, as we see, the population was pretty stable up to 18 hours, but between 18 and 24 hours, the population grew by more than a log. Very similar for Listeria monocytogenes, population grew more than a log between 18 and 24 hours. Performance criteria, here was less than 1 log growth, and the result showed more than 1 log growth. A result for Staphylococcus aureus are presented here. This organism also grew in the product, but the performance criteria, again, it was less than 3 log growth from the initial level. When we look at all of the results and we want to draw a conclusion for this, result of this challenge study, we conclude that performance criteria was not met. Therefore, temperature control for safety is required for this item and cannot be stored at room temperature for 24 hours. The last example that I'm presenting here, it is for a plant-based beverage during batching. This is not the finished product, this is in-process product. Overall study design, target organism, Bacillus cereus and Staphylococcus aureus. And Martha mentioned that these are toxin-producing organisms. They may, grow and produce toxin during batching, and toxin is stable and possibly will not be inactivated by UHT process. Therefore, it's important to study the behavior of these 2 organisms during the batching process. Initial level of inoculum, if you're looking at growth studies, started about 3 log CFU per gram. Storage temperature varies. I will cover that on the next slide. Sampling time, again, 7 to 8 sampling times over the duration of the study. Number of replicate, 3 samples, and 1 trial was completed. Target organisms, Martha mentioned, I just mentioned, they are Bacillus cereus and Staphylococcus aureus. What I am presenting here are the results for Bacillus cereus only. The experiment was done at 3 temperatures, at 25, 15, and 10 degrees C. As expected, the growth rate of Bacillus cereus was faster at higher temperature. As we see, growth rate was faster at 25 degrees C versus 15 versus 10. Let's remind ourselves what was the performance criteria for Bacillus cereus. It was less than 3 logs of growth from the initial level. The initial level of inoculum was 3 here. You see at time 0, the population that we inoculated the sample was 3 log CFU per mil. And then if we look at the 3 log rows, we are looking at the population of 6 log CFU per gram. As here is shown by 2 red lines for 3 log growth. And then the time that's required for each one, at each temperature, the population reached to 6 logs, here is presented by X. When we look at the time required for 3 log growth at each temperature, let's analyze them each line or temperature separately. At 25 degrees C, the population reached to 6 log CFU per mil, sometime between the 2 sampling times of 8 and 24 hours. Here, if we consider this is linear, sometimes around like 12 hours we reach to that, 6 logs. But all we can say based on this data is the population reached to 6 log CFU, sometimes between 8 and 24 hours. At 15 degrees C, population reached to 6 log CFU per mil, sometime between the 2 sampling times of 32 and 48 hours. And at 10 degrees C, the population reached to 6 CFU per mil, sometime between the 2 sampling times of 56 and 72 hours. What is the application of this result? The result of the challenge study was helpful to determine the hold time at various temperatures during the batching step to prevent the growth of Bacillus cereus. Based on the result that we just presented, the batching step may allow for growth and toxin production of the pathogenic organisms. Therefore, we can set the limit for batching step time and temperature to prevent the growth of pathogen and toxin production of the organisms during the batching system prior to UHT process, if this is the beverage that we covered here. And thank you very much for your time and attending the presentation. Scott?
Yeah, thank you so much, Sharon. Those are excellent examples, very detailed. It looks like we have had a few questions that have come in while we were talking. So I would like to remind everyone, feel free, continue to send any questions you may have. We'll get to what we can. If we don't address any other questions that you asked here, we'll be sure to follow up with you directly after the webinar. So I guess the first question I have on here is, and I think it relates to maybe your last question or your last case study there, Sharon. When you talked about that one, you were just looking at Bacillus cereus, Staph aureus. What about growth of other pathogenic or spoilage organisms in that in-process product during the batching? Do you have concerns about that, or do you not look at that when it comes to those challenges?
Yeah, I can cover this one. Yeah, so one of the concerns you might have in terms of spoilage organisms would be that if you are getting growth of spoilage organisms during that batching step, and then you have a thermal process later on, you can run into a situation of incipient spoilage, meaning that these organisms are actively growing in the batch. Maybe they're changing the pH, the quality of the product. You might get separation or off colors, odors, flavors. So yeah, that can still cause issues, but you may not even be able to recover viable organisms. So that would be one issue. And then in terms of pathogens and spoilage, if you're getting that really high level of active growth, sometimes your thermal process may be a little bit borderline, or just may not be enough to address those high levels of organism. So in that case, the high load of organisms may overload the system, and it may not be sufficient to kill all of the pathogens or spoilage and cause issues later on. So yeah, those are two things to think about.
Perfect. Very good. Thank you. All right, so I'll have another question here. Let's see. Can we use modeling to determine the time and temperature for batching?
You're muted, Sharon.
Yeah, Sharon, sorry to interrupt, but I believe you muted yourself.
Okay, thank you. Modeling is a very good and useful tool to predict the behavior of organism, in different matrices with different pH and water activity, for example. It has an application here to kind of like predict the behavior of organism, but not necessarily the modeling can replace the challenge study. Challenge study is still highly recommended because the behavior of organism in the product, might be different from the modeling. Maybe modeling, some of these products are novel ingredients, and maybe those, organism even grow faster in the matrix that you're testing. The other way is also possible. Maybe, this modeling is for broth and adjusted pH. And when you're testing your product, maybe organism grow at a slower rate. Again, it's a good tool to basically predict, but doing a challenge study, performing a challenge study is highly recommended in your product with the target organisms.
Very good. So, and it's kind of applying that to your last case study then too. I mean, you had tested that plant-based beverage at 3 different temperatures. So if someone were to look at that, or I might look at that and say, well, what about 4 degrees C? And, you could probably take that data and maybe make some guesses at that. I would imagine you might say instead, no, let's just rerun this at 4C, let's rerun it further and get a confirmation of that.
That is correct. It's extrapolation of this data. It's not recommended.
Right. Okay. Okay. So another question just came in. So how often do you recommend challenge studies?
I can take that. So I think it really depends. If your product still fits the parameters of the original challenge study in terms of formulation, pH, water activity, and if the process hasn't changed, you probably don't need to do new studies. However, if your product is changing, you're changing your formulation significantly over time, or if you're adding new formulations to your product portfolio, you might want to just review and make sure that they're going to be covered by the previous studies. And if they're not covered, then maybe a new study would be a good idea.
Very good. All right, so another question along the lines of the criterion for the challenge study. Where's that found? You mentioned, I don't know if there's a way to pronounce the abbreviation, but the NACMC Foods Manual, is that kind of where you reference, or where some of that information might be defined?
Yeah, those performance criteria are, I pulled those from that potentially hazardous foods document. So those are the ones where I have the little screenshot of the cover of the document. And I can send that out to the list if you like.
Very well. I think that could be helpful. We could follow up with that after the webinar as well.
Sure.
All right. Question: how do you interpret the results of a challenge study if the results of the 2 independent trials that you're running are not consistent?
I can take that. The purpose of doing the experiment more than one trial, if we can formulate a product at the lower, worst case scenario, great, we have that, all of those parameters set at the worst case scenario and around the challenge study. But in many cases, you cannot really adjust the, pH and water activity in the lowest, or the worst case scenario condition, highest pH, lowest water activity. In this case, it's recommended that the experiment may be done in 2 or 3 tries to capture the variability from sample to sample, from batch to batch, and also, study how different pH and water activity, if sample from sample has a different pH and water activity, how that's going to affect the growth. But a short answer to your question will be if the results of different tries of the challenge study are not similar, the worst case scenario will be considered, and that will be used to decide if the product is TCS or not TCS.
Very good. Very good. Thank you. Okay, I'm not seeing any other questions come in, so I suppose we'll go ahead and wrap up our Q&A session. Once more, I want to remind everyone, recording of the webinar along with a copy of the slides will be available to you 3 days, within 3 days, I should say, of the completion here. Again, I want to thank you, Shirin, Martha, for expertise and presentation today. Thanks.
Thank you.
Thank you, Scott.
Thanks so much. Thanks you everyone for attending. I guess we'll see you next time.
Thank you.
All right.
Thank you.